is Wasajira Annette. I have the red number 2321 Alphabetically, I'm number 8. And I'm going to represent about principle behind agglutination reactions. So, agglutination reaction involves the clumping of particles such as cells or latex bits where specific antigens bind to the corresponding antibodies. And it has this principle, the antigen-antibody specificity, whereby the antibodies recognize and bind to the antigens on the surface of the cells, microorganisms, or artificial particles, forming visible aggregates. So, how the latex agglutination works in diagnostics. So, latex agglutination is a diagnostic test where antigens or antibodies are attached to the latex bits to enhance visibility. So we have the procedures that we use, that those are the latex bits are coated to the specific antigen or antibody, and then the sample containing the corresponding antibody or antigen is added. This might be the blood or urine. If the target is present, it binds to the coated bits, causing visible clumping if positive. And if negative, there is no clumping. So this latex agglutination, it's applied in detection of pathogens such as streptococcus or Neisseria meningitis. So this can be also applied in rapid diagnosis of conditions like rheumatoid arthritis. So another question is like the main types of vaccines and their characteristics. The first is live attenuated vaccine, where it contains weakened pathogens and it also induces strong long-lasting immunity. The examples include measles, mumps, or rubella. And the limitation is like it's not safe to the immunocompromised individuals. The second is inactivated vaccines or killed ones. So these vaccines they contain killed pathogens, safer but may require boosters, and it has examples as polio or hepatitis A. Another example is subunit or recombinant or conjugate vaccine. It contains specific antigens, not whole organisms, safer but with few side effects. The examples are hepatitis B, HPV, or pneumococcal vaccines. So the fourth is toxoid vaccines. These toxoid vaccines, they contain inactivated toxins from the bacteria. They protect against disease caused by bacteria toxins, examples tetanus or diphtheria. And the fifth one is the mRNA vaccines, or it's an emerging vaccine, using mRNA to instruct cells to produce antigens, rapid to develop, high effective. Examples, it's the COVID-19 vaccines. So the fourth question is how hard immunity contributes to the disease prevention. Mainly, hard immunity occurs when a high proportion of a population is immune to a disease or reducing it to be spread. So the mechanism, it protects those who can't be vaccinated, like the newborns or immunocompromised individuals. And it also interrupts the transmission chains, making outbreaks less likely. There are also differences between active and the passive immunization. It's a fifth question, and I made a table talking about active immunization and passive. The first difference, I said, it stimulates the immune system to produce antibodies. That's the active, and the passive it provides the preformed antibodies. So on the active immunization, the duration it uses long term. Sometimes it's a lifelong duration, and passive it uses short term duration. And the mechanism, the active uses via vaccines or natural infections, and passive it uses via maternal antibodies and serum or immunoglobulin. The examples for active immunization is measles, mumps or rubella, and hepatitis B, and passive immunization, antivenom, monoclonal antibodies or maternal IgG. Thank you.